Hello everyone and welcome to today's Biology Daily Booster. This is number 3 in our series for paper 2. It is an advanced information listed topic for those of you doing foundation biology. Higher tier biology is not explicitly listed, but I find it hard to believe there's going to be no questions at all on module 4. So I would say highly relevant for everyone. What we're going to be looking at in our booster today are the different cycles that are involved. Hopefully we do know that we've got a range of different cycles that allow us to basically pass the minerals that things like plants need through animals and then back to the soil again. So we've just got this little loop that goes on for these different chemicals that allow us to have a pretty much constant supply of them. There are 3 you need to know about. Carbon, which we use to make fats, proteins, carbohydrates, DNA. Nitrogen, which we use to make DNA and proteins. And water. So we're going to look at the cycles for those 3 things. Now, the reason that we've got what carbon and nitrogen are used to make is because they can ask that, and they have asked that as previous questions. So just make sure you know at least one thing that's used to make carbon and one thing that's used for the nitrogen there. The first cycle, the one that's probably the least favourite, is the nitrogen cycle. Now the nitrogen cycle is not as bad as it looks on first glance. Yes, that picture looks a bit complex, but not as bad as you think. What we're going to do is we are going to start with nitrogen in the air. So we are starting up here with our nitrogen, which is just N2. So that is our nitrogen gas in the air. Chemistry link, 78% of the air you are currently breathing in is nitrogen gas. Now our plants can't directly take in nitrogen as N2. What we've got to do is we've got to turn it into something else. So what we need to do is change our nitrogen into nitrates. And this is our first bacteria here, nitrogen fixing bacteria. Change the nitrogen, which is just our N2, into nitrates. Now we tend to find these nitrogen fixing bacteria in the soil. Some of them are associated with the roots of plants, but nitrogen into nitrates is what happens. At that point, our plants are able to take up the nitrates, and then they're going to make proteins using them. Which remember, nitrogen used to make proteins. Once we've got the proteins in our plant, our animals may eat the plant, so it gets passed from plants to animals as proteins. And then what we find is plants and animals can both die. At the point of death, then what we're going to find is we go through the process of decomposition, which is our second bacteria, the decomposers. Now, decomposers are going to take those proteins and change them into ammonia. So decomposers, proteins to ammonia. What we then find is that ammonia is going to be changed into nitrates by our third bacteria, the nitrifying bacteria. So we've got nitrates in the soil again, which can be absorbed by the plants. The final type of bacteria in our nitrogen cycle are the denitrifying bacteria, and they just take those nitrates and return them to the air as nitrogen. My suggestion for learning this is learn the 4 bacteria and what they convert into what. The only other point you then need to remember about the nitrogen cycle is that the proteins in plants are eaten by the animals, and that's how it passes along to them. The second cycle, the water cycle. This is basically key terms. We're going to start up here in our clouds. What happens is every once in a while, and we're very familiar with this living in England, then rain falls. That is the process of precipitation. Water droplets in clouds get heavy and fall as rain, or snow, or hail, or all 3 on one day in England, you never know. As that rain reaches the ground, then some of it is literally just going to run off the surface into some kind of water system. Some of it is going to actually trickle through the gaps in soil and rocks in the process we call percolation. So if it percolates, it trickles through gaps. Once we've got that water in the ground, some of it is going to be taken up by plants, by their roots. That water in the plants is going to go through the process of transpiration, and therefore we have loss of water vapour from the aerial parts of the plant, and that water vapour goes into the atmosphere. Another place we get water returning to the atmosphere is through evaporation. So anywhere that water is sitting, if it is nice and warm, then water will evaporate, becoming water vapour in the air. That water vapour is then going to rise because warm air rises. As it rises, it's then going to get into the cooler air, and that means we're going to see condensation. As it condenses, it becomes clouds, and then as those clouds continue to cool, we get back to precipitation once more. So you really just need to know those few key words and what they mean. You can then apply it to any question on the water cycle. Third cycle, the carbon cycle. This one I think is easier. We need to remember that there is a grand total of one process that is going to remove carbon dioxide from the atmosphere. So if we've got our carbon dioxide in the atmosphere here, there is only a single process that takes it out, and that is photosynthesis. So photosynthesis is the only one where an arrow will be going from carbon dioxide in the atmosphere into a plant. We then have three options for how carbon dioxide is returned back to the atmosphere. First one, respiration. So any living thing going back to the atmosphere, that will be respiration. If we've got something like a factory, a car, some kind of industrial thing, then that is going to be combustion. The burning of fossil fuels returns carbon dioxide to the atmosphere. The final one is decomposition. So what we find then is that we do have those three processes that we're going to be returning our carbon dioxide to the atmosphere. Only one that removes it. It is worth remembering though that when we're looking at carbon dioxide levels, it's not a case of these are fixed throughout the day. They actually show variation across that 24-hour period. The reason for that, photosynthesis only happens when light is available. So that tells us that carbon dioxide is actually only being removed from the atmosphere during daylight hours. Respiration, however, the process that creates the carbon dioxide which is released into the atmosphere, that happens all the time in all living cells. So carbon dioxide is released at this consistent rate, but photosynthesis is only removing carbon dioxide during daylight hours. So we get a little shape on our graph, like you can see at the bottom. It's just a series of little zigzag lines, because it's obviously going to be going higher at night, lower in the day, higher at night, lower in the day, etc. Now, we do see on this little graph there is a general trend or pattern that as time goes on, the carbon dioxide level increases. And that is down to us as humans. We are almost solely responsible for that, so yay humanity. The final bit of our little booster today is looking at decomposers and detritivores. When we talk about decomposers, these are microorganisms. So if they ask you to name something that is a decomposer, bacteria or fungi is what they're looking for. They don't expect you to name a specific bacterium or a specific fungus, literally just bacteria and fungi, that's what we're looking for. The way that decomposers work then is they're going to break down this dead organic material as well as waste products like urine and feces. And in doing so, they return those minerals to the soil so they can then re-enter plants, etc. as it goes through the cycles. We do have this term here, saprophyte. Now, fungi, for example, are saprophytes. They feed on dead material by releasing the enzymes onto the surface. So they don't have a digestive system. Instead, what they do is they secrete these enzymes from themselves onto the surface of the dead material. Those enzymes break it down, and then they just absorb those nutrients. So that's what we mean by a saprophyte, something that releases enzymes onto the surface and then absorbs the nutrients. Detritivores are small animals. Now, what they're going to be doing then is they basically feed on this organic material, and in doing so, they're breaking up large pieces into smaller ones. The reason we care about that is because the action of these detritivores means that we get a much larger surface area of our dead material for those decomposers to then work on. Larger the surface area, the faster the rate of decomposition. So just little examples of our detritivores, things like earthworms, woodlice, maggots, all of them work in the same way. They feed on this organic material and break it up into smaller pieces that then the decomposers can work on. We could very easily get the question that's all about usually compost heaps. They love compost heaps, but it's basically looking at the rate of decomposition. Now, we've got several factors that affect the rate of decomposition. First one, temperature. Second one, moisture content. Third, oxygen levels. We want it to be warm, moist, and have plenty of oxygen available. So you've got to kind of look at the question here, because they can phrase it in a variety of ways, basically different versions of compost heaps or compost bins. And you're really just trying to break it down into which of those three factors are at play, and is it actually increasing the rate of decomposition. So what we're looking at first of all is the temperature warm. If it's cold, decomposition is going to be slow. If it's far too hot, then obviously we're going to stop enzymes working and therefore slow down the rate. Hence warm, not hot. We need moisture there, because if it's a completely dry environment, then our decomposers will not be able to survive. And we need plenty of oxygen, because in order to carry out the process of decomposition, they need to be able to respire. And respiration, oxygen is needed there. The only other thing that they could throw in here on this style of question is looking at surface areas. So remember what we said, larger surface areas mean that decomposers can work quicker. So if you've got larger surface area, i.e. you've broken it up into smaller pieces, then you are going to see a faster rate of decay. We could be asked to do a calculation on rate of decay, so that is literally the change in mass divided by the time it's taken to change in mass. That's a nice simple little maths question there. If obviously they're asking you to consider investigations into decomposition, then think about your types of variable. So your independent variable, the thing you are changing. Dependent variable, the thing you're measuring. In this case, most likely a change in mass. Your control variables, anything you want to keep the same. So if we want to investigate how temperature affects decomposition, we need to control everything but temperature. So add the same volume of water using a measuring cylinder. Make sure that we turn the sort of material over once every day so that there's plenty of oxygen available. Think about how you can control the other variables if they ask you how to make this a valid experiment. If there are any of the topics that we've gone through today that you'd like to look at in a bit more detail, we've got the videos on the main channel that you can use. You have your revision guides, I hope, your classwork notes, and there are certainly questions on decomposition on previous exam papers that I do suggest you have a little look through. Don't forget, of course, to join us again tomorrow for our next Daily Booster.